Hi, my name is Benjamin Tesler, and I'll be presenting my work that's titled Non-Invasive Electric Stimulation Using Deep Targeting Techniques Modulates Hippocampal Theta in the Urethane Anesthetized Rat. My project has three main aims, with the first being modeling. I am taking a reconstructed rat head model, importing it into a physics simulator to simulate electric stimulation to optimize where to best place our stimulating electrodes. Then we validate our results using phantoms, which are gel-injected rat skulls that represent what the electric stimulation will do in the rat head. Aim 2 is using what we learned from Aim 1 to target specific brain regions to modulate or change the naturally occurring brain waves. And Aim 3 is using all this to find possible treatments in our rat models of epilepsy, which are our future experiments. What influences neurons, which are the principal brain cell, when doing electrical stimulation is a directional electric field. Electric field is created when there is a voltage difference over some distance. As can be seen in the cartoon of a 2D representation of a neuron with the cell body on the bottom and a large dendritic tree upward, which is basically its receiving cable to receive signals from other neurons. When we place a voltage difference in this direction, we influence the neuron's internal voltage and, depending on the direction of the electric field, either increase or decrease its probability of firing or spiking, which is how neurons communicate to other neurons. The next important thing to consider with electrical stimulation is targeting this electric field to points of interest for our desired effects. When we consider a cube being stimulated with an electric current injecting and returning on entire faces of the cube, as can be seen in the left image, we see a uniform electric field over the entire volume, which is shown as the arrows being the same strength and color. However, if we use electrodes that are smaller than the face of the cube, as can be seen in the right image, we see a steep drop-off of electric field intensity as we get further, deeper, and away from the electrodes. This causes challenges because when we attempt to target neurons that are deep, we are compromising either stimulating neurons that are closer to the surface, or we stimulate everything indiscriminately within the brain. Transcranial electrical stimulation can be a very valued tool for research and in the clinic due to its low cost, ease of use, and relatively low risk. If proved to be effective, it can be widely deployed around the world for various purposes and treatments at a fraction of the cost of other methods and avoids the risks and inconvenience of invasive surgery. The two techniques of transcranial electrical stimulation that I'll be discussing are the conventional high definition and the temporal interference technique. The conventional method uses two large saline-soaked sponges, one termed the injecting current electrode, is placed over the region of interest that sends in either a constant or alternating electric current. The other, termed the return current electrode, which is placed far away from the region of interest. A more advanced version of this is termed the high definition, where the electrode sizes are smaller and the return current electrode is split into multiple electrodes, so the intensity underneath is also split in order to avoid stimulating regions that aren't of interest. The other technique is temporal interference stimulation. This uses two pairs of electrodes, each sending higher frequency current with a small frequency difference. The resultant summed electric field have a wave envelope or beat frequency at the frequency difference. The amplitude of the beat frequency is largest along the midline between the two closest electrodes of each of the two pairs. It is assumed that neurons aren't affected by the higher frequencies, termed carrier frequency, due to the neuron's low pass frequency filtering properties. But neurons are influenced by the beat frequency due to the neuron's rectification properties, which are when the neuron allows in electric fields only in a certain direction. The conventional and high definition cases have over 10,000 publications and have been a hot topic of study, with its low cost at about $100 and its uses being wide-ranged, such as for neurodegenerative diseases, mental illnesses, epilepsy, addiction, migraines, chronic pain, stroke, and learning disorders, and many more. And the temporal interference is a more recently development, having only about 500 publications and being similar in cost and use cases. Here we have a simulation example from each of the two techniques. On the left we see the injecting current electrode in blue and the return electrode as a ring in gray. Below we see three orthogonal planes of the electric field distributions as a heat map, with red being the high intensity and blue being low intensity. On the right, we see temporal interference, two pairs of electrodes with one of the pairs injecting electrode being blue and the other pair's injecting electrode being red, with the return electrodes both being gray. By a quick glance of the electric fields, we see that the temporal interference is much better at penetrating high-intensity electric fields deep into the media, which makes temporal interference possibly a better technique for targeting deep structures in the brain. To quantify the penetrative effect of various techniques and electrode placements, we're using a parameter called spread. To calculate spread, we take the volume at which the electric field is greater than half of its maximum, termed V half, divided by the depth at which the electric field is equal to half of its maximum, termed D half. To visualize this, we are looking at the electric field heat map of a cutout of the cube to the left. The cut portion is the purple prism within the cube. On the right, we shaded everything red that is greater than half the maximum and shaded light blue everything that is less than half the maximum. V half would be the volume that is red, and D half is the length of the yellow arrow. Therefore, the electric field distribution with the best penetrative effect would have the smallest spread. Here we are looking at a D half by spread scatter plots, with each point being a different electrode paradigm. As can be seen in the top left plot, which is the temporal interference case, as the distance between the injecting electrode gets larger, the D half and the spread gets larger. For the bottom left graph, the conventional cases, as the injecting electrode radius gets larger, so does the D half and the spread. When we compare the two together, we see that for the same D half, the temporal interference cases have a significantly smaller spread, showing quantitatively how much better temporal interference could be for targeting deep structures in the brain. So why is the hippocampus our target in the urethane anesthetized rat? First, it tests our ability to target deep. Second, under urethane in the rat, the hippocampus generates spontaneous brain rhythms, theta, which is when populations of hippocampal neurons are firing in unison. 3. Theta is an important brain rhythm involved in learning and memory, navigation. And 4. Theta is possibly anti-epileptic. So our experimental design, we have two groups. The first one is the temporal interference, where we stimulate for 1.5 seconds at the 4 to 5 hertz beat at 3 kilohertz carrier with 3 various amplitudes at 800, 1200, and 1600 microamps. Group 2 is the sine wave stimulus, where we stimulate for 1.5 seconds between 4 to 5 hertz with amplitudes of 5, 10, and 20 microamps. And each of these are randomly ordered with about 5 seconds in between each stimulation. Here we are seeing images of 3 different angles of the electrode montage for our stimulation. We have 2 pairs of electrodes, blue and green, and a recording electrode probe in yellow. As can be seen, we are stimulating the contralateral hippocampi to avoid stimulation artifacts that would affect our data. We put in a 3000 and 3005 Hz label and the positive sign to show an example of how we are stimulating and which electrode is our injecting current electrode, respectively. In the sine wave stimulation group, instead of using those two higher frequencies, we are only using a single frequency within the 4 to 5 Hz range for both electrode pairs. Here we have average spectrographs, which are heat maps with time on the x-axis and frequency on the y-axis. When we see the image contain pixels that are more yellow, it means that the amplitude at that frequency is large. The rows are the two groups, sine wave stimulation and temporal interference stimulation, and the columns are the intensities with low being at the left and high being at the right. As can be seen in the top row, as the intensity gets greater, there is a larger amplitudes only when the stimulation is on, as can be seen as the green highlight at the bottom between 1.5 to 3 seconds. For the temporal interference group, we see that not only does the amplitude get larger during stimulation, but has a lasting effect for several seconds after the stimulation ends. Here we have an example of one of the sine wave stimulus at 20 microamps. The top plot is the stimulus waveform, where the green highlight is the start and stop. The second from the top plot is the raw data with its filtered data overlaid. The bottom color highlights are when theta is determined to be on, and its associated frequency with the color-coded legend is to the right. The third from top plot is the extracted amplitude, with the dashed line being the threshold we use for detecting theta, and the bottom is the spectrograph. As can be seen, the sine wave increases the amplitude only during stimulation. Here we see another example in the same format but with temporal interference stimulation at 1600 microamps, as can be seen in the top plot. What we can notice is that this turns theta on and is sustained for the entire length of the example. Here we have histograms for the temporal interference for the percentage that theta is on for various time groups of 1.5 seconds, before stimulus, during stimulus, and after stimulus. As we can see, theta is completely on at a rate of about 60% of all stimulation sessions, which serves as our baseline. However, it is slightly increased for the 1200 microamp group during and after stimulation, and for the 1600 microamp group, the increase goes from 60% before stimulation to about 90%, meaning that for most sessions that theta was off before the 1600 microamp group, it was turned on and stayed on after stimulation. Here we have histograms of onset and offset times of theta for the temporal interference group. We have theta onset times in the top row and offset times in the bottom row, and low intensity stimulation on the left and high on the right. We can see in the top right plot that there is a large probability that theta turns on about a half a second into the stimulation, and in the bottom right plot we see that there isn't a large probability of any offset of theta. Here we have the same histogram format for the sine wave stimulus group. As we can see in the top right plot, a large increase in theta onset at stimulation start, but also a large increase in the probability of its offset when stimulation ends, as can be seen in the bottom right plot. All this data shows that temporal interference seems to be better at targeting and influencing theta than the conventional method, as it can elicit long-lasting theta. Being able to elicit long-lasting theta compared to just an increased transitory theta can give us a much larger therapeutic effect when translated to the clinic, and can give us better tools for understanding the brain. Also, this can serve as an example for being able to target other deep structures or other brain rhythms with more precision than current methods. Thank you for your attention. Email me any questions, benjamin.tesler@downstate.edu.